Okay, so to learn about enzymes, first thing we need to know is that an enzyme is what we call a biological catalyst, and a catalyst speeds up a reaction. So an enzyme speeds up all the reactions inside your cells. So an enzyme is made up of proteins, and it has a specific shape, and we have two different models that we look at. So we have a lock and key hypothesis, and we have an induced fit hypothesis. So the lock and key hypothesis says that we have an enzyme that has a specific shape that we call the active site. So that's going to be our enzyme. And in that active site, we fit a substrate. So now the substrate fits into the active site perfectly without any adjustment, so that your enzyme can work on it and break it down into products. So that is then our substrate. So in the lock and key, nothing has to change, just like when you put a key into a lock. So you place it in, you turn the key, and you open the lock, and when it comes out, your enzyme remains completely unchanged as it started. And then the next substrate can go in, fit perfectly, and then be used again over and over and over again. But with the induced fit hypothesis, we have the enzyme that has its active site, but when the substrate gets put in, the substrate doesn't quite fit perfectly. And what happens is the enzyme slightly changes shape around our substrate so that it can fit perfectly. So whereas sometimes you might end up in the lock and key, it might not be perfect. In our induced fit, we can change that shape so that it will fit perfectly. Then it will change back, release our products, and be ready for the next one to go in and out. So in the induced fit, the active site changes shape around the substrate to get a perfect fit. In the lock and key, our substrate just simply fits into the active site as it is, so that we can then work and create our products. So if we look at what we call the mode of action, that is how enzymes work. So we can look at how enzymes work by first looking at a graph of energy versus a progress of a reaction. So a reaction is going to take place and it has to have a certain amount of energy. So what's going to happen is we are going to have reactants that are going to react together at that energy, and when they react, they need to overcome a certain amount of energy before they can become products. Like that. So this amount of energy that we look at, that's how much energy is needed for a reaction to take place. And we call that energy, so from where our reactants start up to where it needs to be, we call that the activation energy. So without the activation energy, a reaction can't take place. Once that activation energy is reached, then we can form products. So what happens with an enzyme is that it lowers the activation energy. So a reactant can react with each other, and instead of needing to go all the way up here, we can end up only needing a shorter amount of energy to become products. So this is then going to be the activation energy with our catalyst or our enzyme. So we're going to lower that activation energy in order for a reaction to take place. So what's going to happen is we have an enzyme which has an active site, and a substrate that it's working with. That substrate is going to move into the active site, and we are going to end up with one molecule that has an enzyme and a substrate in it, and we call that an enzyme substrate complex. So this enzyme substrate complex that gets formed is where our activation energy is going to be lowered so that it becomes easier to react and break up the substrate into products. So then what's going to happen is, once we've lowered the activation energy, our enzyme stays the same. And our substrate has then become products, which get released. And the enzyme can go back and be reused again to make a new enzyme substrate complex. So that's our mode of action of how an enzyme actually gets together and works when it is catalyzing a reaction. So now we can look at the effect on enzyme activity by different things, starting with concentration of the enzyme. So enzyme concentration is how much enzyme we have. And what we can do is, if we had to plot a graph of a reaction happening over time and how much product we have, I would have starting at 0, increasing my amount. As we go, I get more and more product. If I had to increase the enzyme concentration, I am going to have more product created over a shorter amount of time. And if I changed my concentration again, I would have even more. So as the concentration increases, my graph would get steeper and steeper and steeper because I'm going to make more, because there is more enzymes active sites available to be able to work with. So if you have to take each of these at their initial, we can plot a graph of our initial rate of reaction over enzyme concentration. So as I increase in enzyme concentration, if I had to take each one of these initial rates and plot them on that graph, what's going to happen is I end up with a straight line, meaning as the enzyme concentration increases, I become proportional in terms of increasing rate of reaction. So when you increase your concentration of enzyme, you increase your rate of reaction. If you look at substrate concentration, so that's the thing that your enzyme's working on, as you increase in substrate concentration, your initial rate of reaction is going to increase, just like with the enzymes. But it's going to increase until it levels off. It will always have this shape of increasing, and then it's going to end up going straight. Because at this point, where it levels off, we call that the V max value, because that is where we are then limited by the amount of enzyme that we have. Because once all of the active sites are full, you can't have any more active sites that are being filled. So that is the maximum amount that you can make. No matter how much more substrate I add, I only have a certain amount of enzymes. It's going to be a limit, what we call a limiting factor, because that's then going to prevent any more active sites from being filled once they're already full. If we look at a temperature graph, so our temperature for rate of reaction, as we increase in temperature, our graph is going to go from low. It's going to increase the rate of reaction to a point, and then it's going to decrease rapidly. So this point, where it's the highest, that temperature, is what we call the optimum. Once we go past that specific temperature, it gets really, really hot, because this is temperature from 0. Let's say this is 40 and 50, etc. So once we go past and we start to increase the temperature even more, what happens is our molecules start moving faster. And when our molecules move faster, they start to break the bonds in between them. And when they break, we end up with a denaturing happening, because it's broken all the bonds of the enzyme and the active site loses its shape. So when our active site loses its shape, it can no longer take the substrate and your enzyme can't work anymore. So on this side of our graph, where we have this rapid decrease, we say that's where it becomes denatured. On this side of the graph, where we have low temperatures, it's not denatured because the enzyme is not breaking apart anymore. All we're doing is lowering the temperature, so molecules start moving slower at a low temperature. And when they move slower, they don't break, they just become inactive. So they've just slowed down to a point where they can no longer work properly, but they can work a little bit. And if we increase the temperature, we would get back to optimum and our enzyme would work fine. So on the side in the cold, we get inactive, and in the hot, we get denatured. And then for pH, every graph, if we look along, every enzyme has its own specific pH, and we have a thin graph around that specific pH. That point where we have a high rate of reaction is the optimum pH, just like optimum temperature. But now what pH is pH is because of hydrogen ions. So any pH that we have in there is because of the H plus ion concentration. Okay, whenever we see square brackets, it's an abbreviation for concentration. So a low pH has a high concentration of hydrogen ions. A higher pH has a lower concentration of hydrogen ions. And hydrogen ions will interact with the bonds of the protein structure. So the more hydrogen ions you have, it starts to affect all of those bonds in the proteins, and specifically in our tertiary structure. And once it starts to interfere with that, it's going to start to break up that active site, because the hydrogen ions are starting to bond instead of bonding to the amino acids inside the protein. So what we need at an optimum pH is we need a substance called a buffer. And a buffer will keep the pH at there so that we do not have this denaturing as we change pHs. So a buffer keeps the pH constant. To help our enzymes work correctly, because there's a very, very narrow range around pH where our enzyme concentration's going to stop working. So those are all the different effects that we can have on enzyme activity. So we have enzyme concentration, substrate concentration, temperature, and pH. And we need to know the specific shapes of each graph and what happens when something is denatured or inactive, or when we're going to increase in concentration or decrease in concentration.